Oh, it really wants to go in that box.
Oh, wow.
I dropped the target on that at 1833.
Yes.
Hold it on
got
top
it.
of the
Thank
lid,
you.
open the box, and then swipe it in. Yeah,
33.
Yeah.
That scene from Happy Gilmore where he's trying to get the ball into the hole.
Go into your home.
Yeah, that's your home. Don't you want to go home?
Well, I feel good that we are deeper and we're still seeing cool biology. Dive.
Good biology.
Okay, Carrie.
Open the drawer a little.
I don't know if that's going to help. I mean, it's not going to...
Maybe I'll just float right in.
Not like the lid's going to pull back. I got to slide it.
No, come
this
back.
is ridiculous.
Yeah, okay, let's not do that at this point. I guess you can take another piece.
Okay, zoom in.
If you guys really need it, you do, right?
Zoom in.
And Kirk Sato on Instagram is asking if there's anyone we know studying brittle stars and climate change.
Okay, where would you like the piece from?
The same place. Oh, from where from? Wherever you can get it, I think.
Like here?
Yep. So the answer to Kirk is I don't know. I don't know anybody studying brittle stars and climate change, but a lot of people are studying climate change, so I'm guessing somebody might be.
There's a potential project right there then.
Ooh, brittle star.
So Greg, do we need to try to get the Acesta?
Is this
We really need to make a little headway on the dive. We haven't gone anywhere yet, so.
Is this going in the same bio box?
Yes.
Yes, please.
Ready for tool tray out?
Yeah, do it.
Is there a fear of the
Yeah,
other guy coming out?
Fear.
More?
More.
Do you happen to get photographs of the Acesta clamps?
I did. Yep.
Probably.
Sorry.
Okay, and
Okay.
there we go.
Those are up.
That's I would just give that the same sample number.
Yeah.
Okay, what are we doing now? Are we moving on?
Moving on.
Yes, please. So no rock or anything around here? Oh, did you want, Kira wanted to look at the rock. Did you think we should
We can look.
pick one up?
Okay, I want to get off of this spot here.
Nice sampling, Bob.
Yeah, that was an amazing bit of piloting. I don't think I was breathing during that entire time.
All right, what rock we want here?
If we can get something that's near the outcrop, or maybe some of these look
You want
okay.
something that's broken off from the big rock.
From the,
Like, are we on the face right now, or are we on the top of it?
we're on top of it.
I don't think so. Of the ridge?
Of the thing where the sponge was.
Yeah, sorry, that's what I meant, the smaller feature.
Not sure what smaller feature.
So you want something off of this?
It would be interesting if there is anything, but maybe there's not. Maybe it's hard to sample. Basically, for these crusts to get thicker, they just need to be sticking out somewhere where the currents are a little bit stronger and they're not as sedimented.
Do you see anything? I can point if.
I can't really see anything now. Yeah, maybe up a bit higher. I definitely don't want to get into the really sedimented part below it. Maybe up to the left, near that little trail of stars. There's one on top on
Okay.
the right of the screen now, below that rightmost brittle star.
Yeah.
The blackest looking one.
Okay.
Here?
Just below that.
The two right next to each other?
Yeah.
These?
Yeah, the more forward one.
This one?
No, closer to us, sorry. The one that's more black.
Or aft?
Yeah.
Can you zoom in?
This one.
Yeah. It's got a nice bumpy surface, which we call botryoidal. That's a signature texture of ferromanganese crusts. So I think it looks pretty good. Looks like it'll fit in the bio box.
Yep.
So this one, you want us to try and keep upright without spinning?
Yeah,
Yes, please.
Yes.
Thank you, Erin, for that reminder.
Zoom in.
Couple of grabs, please. Thank you.
Yep.
Yeah, I think there's a little bit of crust on there. Hard to tell the thickness without cutting through it, but it's got a good texture. When they're really fresh and completely unsedimented, the surface textures are just incredible. But in the borderland, we haven't seen anything like that.
Little
Got some
ophiuroid on it.
worms. Okay, we'll keep it.
Okay. Yep.
Put it in the bio box.
Which
before
one?
we lose anybody.
Let's choose one.
One of the starboard boxes, if you reach.
You're trying to keep the critters on it.
Yes, we're trying to keep the critters on.
So I gotta turn off that thruster.
SVCMD.
See, yeah, SV.
Okay, ready now?
Yeah.
Off. Do you want to fill the back or the front first?
Probably the back, the starboard. Aft.
Aft?
Oh, no, aft is forward. The
Oh,
other.
aft is back.
Aft is back.
Aft is back. Okay.
Aft is after. Towards the back.
So we're looking at one blue?
Yeah, how about aft inboard, one blue? Ready?
So
Not
that
yet,
will be rock
not yet.
one.
Raj,
Okay.
R1.
Okay.
Sample tray out.
All right.
I
Okay.
feel like we lost Sam.
Still here,
What's the-
just
Oh,
waiting
aft
for
inboard.
samples to wrap up.
Thank you.
inboard.
Yeah.
That one.
So that's one blue.
Yes, one blue. Sample NA124 R1 into one blue.
Bob, I was seeing a
Sample
lot of
tray
breakup
in.
when you
It sounds
actually,
so
Yeah.
official.
So what was that on?
seeing it on all your SD cameras, really.
All the SD cams?
Yeah. There's starboard bio, brow cam. I think you got a little bit in your Zeus too.
You want the vertical thruster back on?
Hang on. Okay.
Yes?
Yeah. Do it.
Okay.
If we could check some
Back
light,
on.
if we could check some light, we may be able to see something.
All right. I think we should be coming up on the steepest part. Mark leg suggests that might be a little further east. We'll continue along our chosen track.
Can we give Bob some props
Right.
for that sampling? That didn't look easy.
Nice
That
job.
was awesome.
I think we're going to have lots of steepness today.
I need a bio break, though.
Okay, I'll
Oops.
take over.
Still seeing a little bit of
We're just
flicker.
transiting?
Until
Never mind.
they shout, yeah.
Okay.
I can't actually go anywhere. 'Cause I got
So I
that
should,
too.
oops.
Are we starting a transit? Is that what I heard?
Yeah, Bob just has
Bio
to
break.
have a quick
Okay.
break. Quick, like lightning. Yep.
Well, as we're waiting, we have wrapped up an hour, shockingly. So one more question before we go. Folks are wondering what is happening to these samples once they're back on board Nautilus, and then what will be the next steps once they're back into labs across the country.
Oh, that's a great question. Well, okay, let me let Devin describe what we do with the rocks, and then Gabe can describe what happens to the sponge.
Right, so our lab is focused on the macro and myofauna we're going to find on these rocks, so the little creatures on them. So what we do is once Herc is back up on deck, we're going to take the bio boxes out with the rocks in them. And then we're going to take the rocks out, put them onto little trays where we'll then take our little forceps tweezers and pick up all the
Photos
first, of course. And then we'll let the microbio folks do some swabs on the rock before we touch it. And then once they're done swabbing it, we're going to pluck all the little critters off of it. And then we're going to sieve the water, 300 micron mesh sieves and 42 micron mesh sieves to filter out the macro and myofauna before we process them back at the lab at Scripps.
Right. We sort the samples under microscope and pull out the small invertebrates that were both pulled off the rock, but also that fell off the rock into the containers that we're collecting them in. And then the rock gets dry. Well, the rock gets dried and it's given to Kira, and she could tell you what she's going to do with it.
Yeah, on the day after the dive is when I finally get to do my ID, because although rocks look like rocks when you see the outside, because we're at a water interface, there's always a bit of an alteration at the surface due to the interaction with water. So the first thing I do is scrub off all the gross biology. I think it's gross. Any sediment or remaining goo that they didn't take in their quantitative analysis. And then I cut it on a rock saw that I brought on board. The crew here was nice enough to give me a table out on the deck where I can use my wet rock saw that has a diamond blade to cut the rocks in half so that I can get a clean face to see what's the inside of the rock and start to identify if it's a ferromanganese crust, and if it is, what kind of rock it's been growing on, and then also ID the phosphorites and give the biology team the first clue as to what the associated bio has been living on. And then I will pack them up and take them home to do a series of mineralogical identification with X-ray diffraction and or Raman spectroscopy. And then I'll grind them up and dissolve them in acids and analyze them for their metal content. For pretty much every element in the periodic table, we'll try to figure out the concentration in these rocks.
Hey, if we could begin a transect. Here.
What should our heading be? 053. Okay. Thank you.
Great. And then
All right, sorry, so yes, you'd like to start a transect as we get moving?
yes, please.
Okay,
All right, this should be transect 1.
okay. Just wait for them to get on heading and then we'll drop a target.
All right, well, thank you so much, everyone, for answering some of those questions from the viewers today. For folks watching on nautiluslive.org, this expedition will be wrapping up on Friday. So hope you're able to explore with us for the next couple of days and
There's some sea cucumber.
continue to explore with us for the next month as we continue seafloor mapping until our season wraps up in December. Thanks, everyone in the van.
Thanks,
And,
Sam.
Thank
take
you.
care out
Thank
there.
you. Thanks for all your questions.
Thank you.
No,
Sorry, Gabe.
I wasn't on.
Get it.
What did you need?
Nothing.
Oh, okay.
Nothing.
All right, so we're on heading. So the slope is gonna keep going, right? You ready? You prepared?
Yeah.
All right, let's do it then. Bridge nav.
Bob?
No dropouts on the optical. I don't know.
I just briefly lost control of Zeus.
You've got Argus, yeah?
Can we get a...
What is that?
What is that in Argus
Oh,
here?
standby. Is everything okay?
I'm
Can
okay.
tell it might be a jelly.
I just heard something. Okay.
I thought it looked like a turtle.
Bridge nav, 10
think it
meter
is,
step
though.
bearing 053.
I don't know if a turtle would be this deep.
Yeah.
Yeah, I don't think so. It just looked like it for
Oh.
a sec.
That looks like a jelly.
Yeah, it looks like a jelly.
A jelly.
It seems to be attached down there, maybe. No, it's drifted by.
Gabe, we didn't get to hear what your lab does with the little swabs you take from the rocks.
True. Yeah.
Oh.
Wanna hear? True. Yeah. You wanna hear about it?
Oh yeah.
So yeah, we plan from those swabs, extract DNA, extract RNA, and study the bacterial community in those rocks. Compare them
You want a junk zoom?
from the
Jump
different
zoom,
dives.
please. Thank you.
And then
Can
correlate
you
that
tilt
data
the,
Porch light?
with,
no, I was going to say, can you tilt the Argus camera up a little bit?
I'll turn the porch light off.
With
Okay, that too.
with the
Just
data
saw
you
a
find
fifth cucumber species.
Was that
Purple, the big purple one.
yeah.
I'm
Dark
coming
purple.
down.
Have we officially started yet?
You got to keep it same height off the bottom.
It's tough to
So
go
you-
up and forward and down.
Yeah, but don't go too fast.
Aaron, did we officially start our transect?
Yeah, sorry, I
Just
wasn't
do it
on
in
SBL.
Auto X.
Yes,
Are
we
we in
did.
Auto XY?
Yeah. I don't think
Alright.
so. Am I?
You're in Auto XY.
Well, I've
Yeah,
been-
but just leave it be,
Okay, not
and then
touching.
we'll do it. And then turn this down.
Okay.
So we've never actually transit anywhere, and we're carrying all this dirt around with us everywhere we go.
Okay, do you want to start over?
No, it's just I'm telling you that's why we have all this dirt.
Okay,
We haven't moved anywhere. We keep carrying it around with us.
okay.
Just you got to do that.
Okay.
Oh, actually we have that on too. Okay, get where you like it.
Is that okay? Right here?
Yeah, now let go. And then we keep this way down here.
That's not a
So we got
crinoid bottom right, is it? To the right of the sponge? Is that a stalked crinoid? It's hard to tell from up here.
It's hard to tell.
We're trying to get set up for a transit though.
You have moved a bit, just FYI. We might make an adjustment to the bearing. So Lisa, this transect will take us past AM, our next waypoint. Is that okay with you?
Yeah, AM was just you can tell us when we're there so
Okay.
that we know we're at the top of the ridge.
Okay.
But
Yeah,
Uh-oh.
and Mark is
Yeah.
calling this the fault scarp here, I think.
That happened
Right,
only
so that was
the top of the
recently.
fault
We got a
scarp?
ground fault.
What?
We just
The
had a
top of the
ground
fault
fault there.
scarp.
Well, if that's interesting at all for the seats, I don't know if we missed that part.
I briefly lost
so
Zeus
so
too,
we may be,
or at least
Kara, you can
telemetry
tell
says.
everybody what this feature is then.
Hey, it's
Wait, just hang on a second. Can we just hang on just a moment here?
Yep.
I need to see what's going on.
Bridge
Sure.
nav,
We're
hold
getting
position.
dropouts.
Thank you.
Argus all Tim is weird.
I'm gonna kill that target, and I think we should start over once we're all situated.
We have technical difficulties right now. We have to work out.
Understood. Science row, I'm just letting you know I'm going to get rid of that target.
Okay.
Thanks,
Okay.
Aaron.
Yep. Oh, crap. Did I get rid of, what was our last sample number?
177.
Okay,
Raj 177.
cool.
The last one,
Uh-oh.
no sample numbers for the transect.
No sample numbers on transects.
Uh-oh. Oh, I didn't want that. Where's the Atalanta? Damn it.
Um, 0 ohms.
It's what?
Well, they both said 0 ohms. Is that because you were starting up?
Because we had to restart the top side, so
Okay.
we're all messed up now.
Okay.
You can tell it's been an interesting dive because I feel like
Oh,
she only got to
man.
ask a couple questions. Whereas she got a lot in on the last shift because there was more transiting.
A lot of action going on.
It hasn't come up
EE
yet.
support? That's me. That's me. We need data. I don't know if he's gonna be able to help, but oh, DE. I thought you
Oh,
said EE.
Data. Okay.
Yes. Can you come up, Kylie? Just
Yep.
never mind those stupid readings.
Yeah, of course.
Yeah.
Our delta's 23.
So science, we had a crash to the Herc topside. It's causing some issues. That's what we're trying to straighten out up
Can
here.
you take off your auto heading if
Yep.
it's on?
Yep.
Thank you.
Oops. Okay, so turn it off. Auto head off.
What is going on there?
They're just restarting everything.
I need to get in a position where I can look into it without trying to drive the vehicle at the same time.
Makes sense.
We aiming for a 0 delta, spread out?
Yeah. Yeah, you can just let go.
Leave it? Okay.
Yeah.
So they need to restart the computers.
Mm-hmm. Okay. Okay.
For everyone at home, channel 3 on the quad is showing the Argus butt camera. It's a camera that shows the tether heading out to Hercules, and that bright light, the end of that tether is Hercules. That's what the pilots were talking about a little while ago, stretching out. I want to stretch out that tether so there's no entanglement or wraps in that cable. Of course, you can't hear them on SPL right now because the conversation is between the two of them. And they're highly focused. It appears they have things under control.
So we have a topside back front row. We're just trying to come back down and get situated. Or back row, sorry. Yeah, that's good. Nice clean Herc. We were heading 053 if we can kind of get back in that orientation.
lots of white sponges and brittle stars.
Too bad she wasn't doing the Q&A right now. That would have been a good time for us to sad how that worked out, but
They might do it again.
yeah.
So for everybody watching, we're back on the bottom after solving a few electronic problems. Looking at a lot of broken up ferromanganese encrusted boulders and rocks.
Are those like clamshells on the left?
Ooh, well, there is a clamshell.
Yeah.
That would be interesting. You're seeing more than one?
Yeah, I saw another one further left.
So they could be the acesta shells, but I'd be interested to know if we're looking at any vesicomyid clams. They would be indicative of a methane seep.
Greg is saying acesta is phony. It's phony.
It's
A
can.
can? Yeah.
So just checking in on some of their instruments. They are not confident in some of the readings right now. So standing by.
Raj?
Well, maybe we can, are we able to use the cameras?
Nothing that involves the pilots right now, unfortunately.
Okay. There's a single stalk coral right at the bottom of our view that Steve's commenting on. It's not sure what it is. Could be a black coral.
When we're well situated, I'll try to get their attention and have a look at it.
Where am I going?
You're off to my port side.
Yeah, getting under.
That's a cool ledge.
I don't feel confident at all here.
Okay.
We start seeing the dropouts, I think.
Not good things.
Well, we may bail. 'Cause that's very suspicious. Could you, yeah, run in the,
Yep. Do you just want to maybe just give science a little update on what happened and what we're expecting now?
well, what we saw was some minor glitches in the video randomly happening. And then I suddenly lost sensors, and then my thruster, had no control over the thrusters, which indicated like a topside dropout. And then it went into a continuous reboot mode. That's why we had to have the data come in here, and he just cycled the computer on and off. But whatever caused the initial problem, you know, we're not seeing the glitches now in the video, but it makes me think that we're getting loss in telemetry, and that's bad.
Okay. All right.
So
thanks for the update. That helps. All right. So we'll carry on for now, but we're keeping an eye on things. Yes.
yeah, if I start seeing glitches, like if we start getting dropouts again in the video, then I'm gonna not want to be.
We're gonna reevaluate the situation. Okay. Yeah. All right. All right. Sorry about that, science. We're gonna try and get back on 053, and if you still want that transect, we can start it over once we're all ready.
Oh, my gains reset.
Thanks, Aaron.
Yep. And if I can make an adjustment to your heading to be about 030.
030.
Thank you.
The numbers for gauges and everything, and all of our humidity, temperatures, voltages, everything looks
all good.
Good, normal, yeah.
Okay. And it's a great mystery.
The white sponges are interesting. They're all on the sides of the rocks, not the tops. You have a specific side and a specific orientation.
Were they all downslope?
Let me just check.
Were they all on the downslope side, or did you see any upslope?
No, downslope.
Okay, do you guys need to look at anything here, or we want to get started on a transect?
Is that another crinoid to the left?
Yeah, could you zoom in on the crinoid?
So we're not starting the trans.
No, zoom in on that crinoid, please.
I always forget, so confused. Are we looking for crinoids?
Greg is, yes. He had a special request for crinoids.
You spot them there?
Just might be the same one we've collected.
Okay, zoom in.
Oh, no, I can't tell. That's not a crinoid. Don't think.
What do you think that is?
Hydroid.
Oh, okay.
I think. I am not the expert. But
It's like a brittle star hiding in it.
Yep.
So we don't care about this. Right?
No, we're not.
Okay, setting up for the transect.
Just confirming, do we want to do a transect?
I think we do.
Okay.
Aaron, I'm just curious.
Just sort of
it'll take us off this rocky feature, right, and onto the plane behind.
100 meters from here. I mean, depending on whatever orientation we're at. Saying we went about 03. This is 030. I was aiming towards that, but we can aim it more this way if you like.
I was just trying to get the transect on rocks, and so if we're about to lose our rocks, we don't need to start it. But
It's hard to say because I think we're seeing at least medium slopes in here. It may be more like this and less like the outcrop we just came off of.
okay,
And then when we get towards 100 meters, that's getting us back into the greens, the really low slope. So I suspect that would be completely flat.
okay.
Would it be interesting or possible to do a transect along this more steep slope? I mean, I know it's away from the original waypoint, but
It's possible if that's what you guys want to do.
I mean, we saw a lot of interesting biology and geology, and this is one of our deepest outcropping features. And 100 meters isn't too far, and maybe we would find something to sample before we head upslope. What's our depth here? Is about 1,700.
Our depth is 1708, yes.
I don't know, what do you think, Lisa?
Where are we now? That is the question.
I'm going to zoom out a bit
The
to
ship,
get an overview.
ship is on the steepest slope.
The ship is on the steepest slope according to our slope maps. We could step it up to get you on that contour and
Let's do that.
and then transect along that.
Let's do that.
You want to try that?
Yeah.
Okay. So once I get their attention again, I'll just let them know what the plan is.
And Steve is saying that hydroid we saw was umbellapathes or black coral.
Oh, it was not a hydroid. Okay, we need to scroll back down to, let's see.
All right. You want to hear the story? All right. We're going to just step the ship along 053-ish to get you up into this contour band here, and then we're going to transect along that contour band.
Okay.
Sound good?
Yeah, so we're not doing a transect.
Not yet.
Doing a cruise.
Yeah, we're going to get set up, and then we'll start transecting.
Okay.
Bob, I'm seeing just a little bit of flicker in your brow camera.
Oh, my God.
Sorry. But the other cameras look fine, so it might not be a bad thing. No, starboard bio's flickering too.
So that was not a hydroid,
Port
although
rail.
it was an idarian.
Starboard rail.
All right, so we're going to be going 35 meters at 050. Bridge nav. Can I get a 30-meter step? 050.
Crispin wants to know the name of the swimming holothurians. Nipdiastes? Yes.
Right there.
Another can.
Can you zoom in?
It's like a Coke. Diet Coke.
That one's not old, though. Right?
Mm-hmm.
Maybe it is.
Older logo. And a cucumber next to it?
Clamshell.
Mm-hmm.
It's probably a polykey tube.
So we're aiming for there, but
So,
if anything else comes up that looks nice and steep, we can use that as well.
Cool. Thank you.
Yep.
Jump zoom out.
That doesn't seem right.
What?
That doesn't seem right. The meso for Herc.
What's right?
I said it doesn't seem right, the meso for Herc. Keeping an eye on it.
Oh, yeah, it probably,
Did it get thrown out?
yeah, why don't we, probably upside down, right?
You want to restart it?
Just, yeah, turn it off.
Stop start. The ship moves in, so we're just starting to get a move on.
So that's very concerning because we didn't cycle power to that. It shouldn't have done anything, you know?
Yeah.
So that sort of makes me think there was a power glitch rather than a loss of telemetry.
So should I just hit run here, or is there another
You could
step?
try it. I think we have to flip the head.
Sponge we collected is very common here.
Mm-hmm.
That's good. It's like a dominant thing. It is.
The underlying rock that we're seeing these light ferromanganese coatings on in this region could be metamorphic rock, sedimentary rock, possibly basalts in this region. It's hard to tell until you cut them open.
Yeah.
This whole ridge feature is part of a compression area where rocks are sort of bubbling out of a compression zone. That's why we have this sort of band on the map of a more rugged feature. For our collections, we're hoping
I
for
can't see this at all.
a little bit more exposed, less sedimented areas. In general.
Oh, okay, so the head's pointed in the wrong direction, so. No. Try go down. Although that shouldn't.
Other
I mean, that makes sense now, right?
Sponges.
All right, look, let's just close the program out.
In general, ferromanganese crusts from continental margin regions tend to have a little bit less manganese and a little bit more iron, and the growth rates are higher. And a lot of that is owing to this more sedimented environment than the areas of the open Pacific where we have very low sedimentation rates. If we were diving on a seamount out in the central Pacific,
I hadn't turned it on
you
yet,
would see much less sediment. Lots of really dark black ferromanganese crusts with gorgeous surface textures.
Do you think this sediment's coming from San Clemente Island, or do you think it's coming from the mainland?
I think it's both. I think it's a combination of biological sediments from the high productivity we've been talking about. There could be some sediments flowing off of San Clemente Island and from offshore. And that's another reason why the ferromanganese crusts in the continental margin region they don't have the same metal enrichments as those in the Pacific and the Atlantic, which are considered for their mineral resource potential. But it's important to see how they compare in terms of mineralogy, chemistry, and the associated biology with those more sought after deposits.
Okay. Yeah. So what else is screwed up? so we'll find out.
All right, sorry, we just had to troubleshoot the sonars. I think we're good now. We're almost getting into that contour band, so we'll start our transect moving in the other direction soon.
So that makes me think that was a power issue, not a telemetry dropout.
Sure. Okay.
So if we see funny business, watch the voltages.
Okay.
See, like on the 60 hertz cabinet.
Yeah.
And over there.
Yeah.
Oh yeah? So it was on both, so okay. Yeah. Yeah. But it doesn't take much to send it in a wacky world.
All right, we're gonna just let the ship complete this move, settle out, and then we're gonna be changing heading or
Right.
bearing completely. We'll be going something like 127, 128,
Okay.
maybe 130. So
Hmm.
hold on. Okay, this is, I mean, we're getting into what's showing up as the steepest slope. I don't know if I agree with the visuals that we're getting here. But do you still want to then try and transect along contour here?
Yes, please.
Okay.
I do.
Okay.
Here it is. So
Here. All right, we
we'll
shall
give
do
a
it then.
try.
Thanks, Lisa, for obliging my idea.
So just tell us when we're starting.
Will do. Let me just get set up here.
Zoom in.
Is that seagrass?
Yes. It's like in a perfect circle. Okay, zoom out.
All right, let's get set up for transect.
Are we crabbing along this or?
Yeah, so do you want them to continue to face into the slope and crab along sideways?
Yes, please.
Okay.
I didn't know that was an option.
Well, it's sort of an option.
I'm not sure. It might be harder to
If it's
do
too hard
100 meters.
and we can still see, then
I just don't know about fishing
I
gear and such things.
Yeah, probably should just head on.
okay. Is
Do
this
it-
more like
do it
what
the
it would
normal
look like
way.
if you do it normal?
What was the bearing you want to go?
The bearing we're going to go is 126.
126.
I can just, yeah,
That's
I can do
fine.
it like this.
That's
Is this
fine too,
okay?
yeah.
All right, so 126. Let me get an estimate in for the 100 meters, and then we will get a move on.
Then we'll head back up to our next waypoint.
Kira, I'm going to maybe, we'll see how it goes, but do you mind if we end up changing bearing a little bit to try and stay on high slope?
Oh, yeah.
Doesn't matter? Okay.
We started?
Nope, not yet. I was just putting in some estimate points so I could keep track of where we were. All right, you ready, pilots?
Sure.
Yep.
All right. What bearing did I tell you? I'm out of it. All right. Too many things at once. One, 2, 5. All right. Bridge nav. Can we get a 20-meter step bearing 125? Thank you.
Oh, your target name says 1816 instead of 46.
Oh, thank you. That's like the third time I've done that. I think it's
The
there's
4 is
like,
by the 1.
So what are we looking for, Kira?
Huh?
What are we looking for specifically?
Yeah, we want to see